I want you to imagine, just for a second, a completely bizarre scenario. Let's say you catch a common cold.
Okay.
Just a run-of-the-mill respiratory virus. You get the sniffles, maybe a slight fever, you recover over a few days, but then you realize something incredibly strange has happened.
Like what?
You suddenly possess a completely new, life-altering physical ability.
Oh, wow.
Yeah. Let's say total immunity to a deadly poison, or, I don't know, the ability to digest wood.
Which, just to be clear to anyone listening, remains firmly in the realm of science fiction for human beings.
Oh, absolutely. Pure sci-fi for us. But the wildest part of this thought experiment isn't just the new superpower. It's the fact that you never once interacted with anyone who actually possessed that immunity.
Right.
You never touched them, never shook their hand, never even occupied the same room or city as them. You just caught a virus and bam, you've inherited a total stranger's genetic trait. I mean, it sounds like magic or like a glitch in the Matrix.
When you look at humans, sure. But if you zoom in, way down to the microscopic level, this isn't fiction at all. For bacteria, this kind of wild, seemingly impossible genetic inheritance happens all the time.
It's just an everyday occurrence.
Yeah, actually catching a virus is one of the primary ways they evolve.
Okay, let's unpack this. Our mission today is to understand exactly how that microscopic magic trick works. In this deep dive, we are exploring a phenomenon in microbiology called transduction.
Right.
It's one of the ways bacteria swap genes, part of a broader category biologists call horizontal gene transfer. But transduction stands out from the others because it fundamentally requires a middleman.
Yes, specifically a bacteriophage, which is just a virus that infects bacteria.
And we should probably distinguish it from the other two common methods of horizontal transfer, just so you have your bearings as a listener.
Yeah, good idea. So there's transformation, where bacterium essentially acts like a microscopic scavenger.
Just sweeping up trash.
Basically. It sweeps up naked pieces of DNA left behind by dead cells in its environment. And then there's conjugation, which is, that requires two living bacteria to physically connect to each other.
Like building a tiny bridge.
Exactly. They build a bridge to pass genetic material back and forth.
Yeah.
But transduction relies on neither scavenging nor physical contact. The phage acts as an unwitting courier.
So we are going to figure out how these viruses end up acting as accidental molecular delivery trucks for bacteria. We'll examine the biology of how a virus makes a packaging error, the difference between a random accident and like a genetic hijacking, and we're going to clarify a massive misconception that constantly traps biology students and science writers alike.
It really does.
But I am hung up on the discovery itself because, if you can't see these viruses without a powerful electron microscope, how did anyone in the 1950s figure out they were acting as genetic couriers?
The detective work is actually quite elegant. It happened in 1952 at the University of Wisconsin. A graduate student named Norton Zinder and his advisor, Joshua Litterberg, were running experiments on Salmonella typhimurium. And the irony of the discovery is that they weren't even looking for viruses.
Wait, really? What were they looking for?
They were actively hunting for conjugation, that direct cell-to-cell mating we just mentioned.
Oh, because Lederberg had already proven conjugation happens in E. coli a few years earlier, right?
Yeah, in 1946, alongside Edward Tatum. That discovery was monumental. It eventually won Lederberg a share of the Nobel Prize.
Wow. So naturally, he and Zinder wanted to see if they could replicate that success in a different bacterial species, Salmonella.
Exactly. And to do this, they used a very specific setup involving auxotrophic strains.
Break down auxotrophic for us. That means the bacteria are broken in some specific way, correct?
Basically, yeah. It means they carry a mutation that renders them unable to synthesize a crucial nutrient, usually a specific amino acid. If you place an auxotrophic bacterial strain on a standard nutrient-rich Petri dish, it grows fine because the food is provided for it. But if you plate it on a minimal medium like a harsh environment completely lacking that specific amino acid, it starves and dies. It just can't manufacture the nutrient itself.
So Zinder and Lederberg took two different auxotrophic strains of Salmonella. Let's say strain A can't make nutrient X and strain B can't make nutrient Y.
Right.
Alone on the minimal medium, they both die. But if you mix them together in a test tube and they physically mate via conjugation, some of them might combine their healthy genes.
That was the hypothesis. If they swap the functional genes, you get a new recombinant cell that can make both nutrient X and nutrient Y.
And survive.
Yeah. Suddenly you'll see rare, healthy colonies popping up on the minimal medium. And when Zinder and Litterberg mixed the two Salmonella strains, they saw exactly that. Recombinant colonies grew.
At first glance, you'd pack up and go to the pub. You'd say, great, we found conjugation in salmonella. Write the paper.
You'd think so.
But they didn't stop there. They introduced a piece of glassware that completely upended their own conclusion, the Davis U-tube.
It's a remarkably simple piece of equipment that just forced a total paradigm shift. Picture a glass tube shaped like a capital U.
Okay, pretty straightforward.
Right. But at the very bottom of the curve, separating the left arm from the right arm, is a fritted glass filter. The pores in this filter are microscopic.
How microscopic are we talking?
They are large enough to allow liquid, salts, and tiny dissolved molecules to pass back and forth when you apply pressure, but they are physically too small for a bacterium to fit through.
So it's like taking two groups of people who want to trade blueprints, but you separate them with a towering chain link fence.
That's a great way to think about it.
They can see each other, they can talk, the wind can blow between them, but they absolutely cannot touch. And if conjugation fundamentally requires physical cell-to-cell contact to build that tiny bridge, separating the two strains with that filter should have shut down the genetic exchange entirely.
According to all the known rules of biology at the time, yes. 0 new colonies should have grown from the liquid in that U-tube. But recombinant colonies still appeared.
Which is wild.
It is. The two populations of Salmonella were acquiring new traits, complementing their deficiencies, despite never actually coming into contact.
Okay, I have to challenge this, because if I'm a scientist in 1952, my first thought isn't invisible virus courier. My first thought is, my filter is broken.
Right. Or you might think, well, the bacteria are secreting naked DNA into the fluid, and we are just seeing transformation.
Right, the scavenging thing.
Exactly. So Zinder and Lederberg had to systematically rule those out. They proved the filter was intact. They also added an enzyme called DNase to the liquid.
What does that do?
DNase acts like chemical scissors. It shreds any free-floating naked DNA in the environment. So if transformation was happening, the DNase would have destroyed the blueprints before they ever crossed the filter.
But the gene transfer still happened. The blueprint survived the journey across the chemical scissors.
Which meant the genetic material was being protected by something. It was inside a physical casing small enough to pass through the filter, but robust enough to shield the DNA from the enzymes in the liquid.
Wow.
And that something turned out to be Bacteriophage P22.
The delivery truck.
The delivery truck. The virus was infecting strain A on one side of the tube, replicating inside it and bursting out. These newly minted viral particles were tiny enough to easily wash through the filter and infect strain B on the other side.
And occasionally, very occasionally, instead of carrying lethal viral DNA, they were carrying a payload of host DNA.
Yep. Zinder and Lederberg published this finding in the Journal of Bacteriology in 1952, and they coined the term transduction.
It's brilliant detective work, but it immediately raises a massive mechanical question for me. I mean, viruses have been evolving for billions of years to be efficient replicating machines. How does the wrong data get packed into the viral envelope in the first place?
A good question.
Why is the machinery making such a fundamental error?
It really comes down to the sheer speed and chemical reality of viral assembly. Biological machinery isn't sentient. It doesn't know what it's packing. It just operates on chemical affinities and shapes.
Right, it's blind.
Completely blind. And this brings us to the actual mechanisms. And there are two mechanistically distinct flavors of transduction. Conflating these two is incredibly common, so we really need to draw a hard line between them.
Okay, let's do it.
The first is called generalized transduction, which is fundamentally a packaging error during the lytic cycle.
The lytic cycle, that's the aggressive phase. The virus lands on the bacterium, injects its DNA, hijacks the cell's internal machinery to build thousands of copies of itself, and then violently bursts or lyses the cell open to release the swarm.
Right. And during that hijacking process, the bacterial chromosome is often shredded into fragments to stop the cell from defending itself.
That's brutal.
It is. Meanwhile, the phage is building empty protein shells called capsids. To get the viral DNA inside the shell, the phage relies on a packaging enzyme. In phages like P22, this enzyme acts like a mindless molecular zipper.
Just zipping things up?
Exactly. It blindly feels around the cytoplasm for a specific barcode, a DNA sequence called a PAC site. When it finds a PAC site, it grabs the DNA and starts aggressively threading it into the empty capsid.
Here's where it gets really interesting, because the capsid is a rigid geometric box, right, like an icosahedron. It can only hold a very specific physical volume of DNA.
Yes, it uses what's called a headful mechanism. The enzyme threads the DNA in until the physical pressure inside the box reaches maximum capacity, which is roughly 104% of a normal viral genome.
Okay.
And then the enzyme simply cuts the DNA strand. The lid is sealed and the particle is ready. But because the enzyme is just blindly feeling for that pack site barcode, it sometimes makes a mistake.
So earlier I was thinking about this like a virus building a state-of-the-art fleet of spaceships to conquer the galaxy. But when it comes time to load the astronauts, the loading crane gets confused and accidentally packs the host's living room furniture instead. Sofas, lamps, a blender.
Yeah.
But based on what you're saying, the crane isn't just confused, it's completely blind, operating in a dark warehouse, relying purely on the shape of the boxes.
That's a vital correction to the analogy. Sometimes a random fragment of the shredded bacterial chromosome happens to fold or sequence itself in a way that chemically mimics the viral pack site. The blind crane grabs it.
Oh, wow.
Yeah, it stuffs the bacterial DNA into the spaceship until the ship is full, cuts the strand, and seals the door. And from the outside, that resulting viral particle looks perfectly normal. It has the landing gear, the terrifying injection needle, the whole apparatus.
But the payload inside is just bacterial furniture.
Exactly. There are 0 viral genes inside that specific capsid. For the virus, this particle is an evolutionary dead end. It can attach to a new cell and inject the DNA, but it cannot replicate. It cannot cause a new infection.
However, for the bacterium on the receiving end, it's a live genetic delivery.
It is. And because this error happens randomly based on stray fragments floating in the cytoplasm, literally any gene in the entire bacterial genome can be packed and transferred this way. That's why we call it generalized transduction.
The random packaging error. Got it. But you mentioned a second flavor, specialized transduction. If generalized is a random accident in the dark warehouse, how does specialized transduction work?
It requires a completely different lifestyle from the virus. Specialized transduction is only possible with temperate phages.
Temperate meaning what exactly?
Well, unlike lytic phages that immediately destroy the host, temperate phages have an alternative strategy called the lysogenic cycle. When they inject their DNA into a bacterium, they don't immediately start building spaceships. Instead, they integrate their viral DNA directly into the host bacterium's own chromosome.
So they just splice themselves in and go dormant, like a sleeper agent.
That's exactly it. They become a prophage. And the evolutionary advantage is immense. Every time the bacterium divides and replicates its own chromosome, it unknowingly replicates the dormant viral code along with it.
Wow, that's incredibly sneaky.
Very. The classic example in the literature is phage lambda in E. coli, discovered in 1956. Phage lambda doesn't just integrate randomly, though. It has a highly specific chemical lock-and-key mechanism.
It picks its spot.
Yeah, it always parks itself in the exact same spot on the E. coli chromosome, called the ATB site.
And what's sitting next to that parking spot?
It's wedged right between 2 specific gene clusters. The gal operon, which contains instructions for digesting galactose sugars, and the bio operon, which contains instructions for synthesizing the vitamin biotin.
Okay.
The viral sleeper agent just hides there, quietly riding along, until the bacterium encounters severe stress.
Stress like what? What wakes the sleeper agent up?
Usually DNA damage. For instance, if the bacterium is exposed to harsh UV light, its DNA begins to break down. The viral prophage senses the molecular distress signals specifically, fragments of single-stranded DNA accumulating in the cell.
It knows things are going bad.
Right. The virus realizes the ship is sinking and triggers a panic mechanism. It rapidly induces its excision enzymes to cut itself out of the bacterial chromosome so it can jump back into the aggressive lytic cycle, build those spaceships, and escape before the host dies.
But in its panic, the excision enzyme makes a surgical error.
Precisely. It cuts imprecisely. Instead of cleanly snipping at the exact borders of the viral code, the molecular scissors accidentally snip a little too far to the left or right.
Oh, I see.
Yeah, it leaves a tiny fragment of its own viral DNA behind in the host chromosome, and in exchange, it drags along a piece of the adjacent bacterial DNA, either the gal genes or the bio genes. What's fascinating here is how restrictive that physical limitation is. Generalized transduction can sweep up any random gene because it's grabbing free-floating debris.
Right.
But specialized transduction can only ever move the specific genes that sit immediately next to the prophage's parking spot because it's a mechanical excision accident.
That stark mechanistic difference is really crucial, and the resulting viral particle in specialized transduction is a hybrid then.
Yes, exactly. It contains mostly phage genes, allowing it to still replicate and infect, but it's permanently carrying that specific chunk of stolen host DNA.
Okay, let's follow the timeline. Let's say our viral delivery truck, whether it's the generalized blind crane or the specialized panicking sleeper agent, successfully flies across the environment, lands on a new recipient bacterium, and injects this stolen DNA. What happens next? I mean, I feel like the host cell shouldn't just passively accept a stranger's genetic code. Doesn't the bacterium have defense mechanisms? Like restriction enzymes that act like security guards to shred foreign DNA.
They absolutely do.
Yeah.
Many times, the injected DNA is immediately recognized as foreign and destroyed by the cell's defenses.
Makes sense.
But occasionally, the incoming DNA manages to evade the security guards. If the new DNA is similar enough to the host chromosome, the bacterium's own recombination machinery will actually splice it into its own genome, permanently acquiring the new trait.
Okay, so a successful transplant.
Right. But there is a third, highly bizarre outcome discovered in 1953 by Bruce Stalker alongside Zinder and Litterberg. It's called abortive transduction.
Abortive transduction, meaning the delivery works but the installation fails?
Sort of. The DNA is successfully delivered into the cytoplasm and survives the security guards. Perhaps it circularizes or binds with protective proteins, but it never integrates into the bacterial chromosome. It just sits there as a non-replicating fragment.
Okay.
The genes on that fragment can still be read and expressed by the cell, but the fragment itself cannot copy itself.
Wait, if it can't copy itself when the bacterial cell divides in two, where does the fragment go?
It can only physically go into one of the two daughter cells. The other daughter cell gets nothing.
Oh, wow.
And when that cell divides again, only one of its offspring gets the fragment.
It's like a strange family heirloom. Imagine a grandfather has a prized pocket watch, but he can only hand it down to one child, and that child can only give it to one of their children. The watch is fully functional, it remains in the family, but at any given generation, only one single individual actually possesses it.
That's a perfect analogy. And you can actually witness this phenomenon on a petri dish. If the transfer trait is something visible, like, say, the ability to manufacture a tiny tail to swim, you get what looks like a tiny trail, a satellite microcolony.
A satellite microcolony.
Yeah, it's a fading microscopic lineage. One cell after another expressing the donor trait transiently, a ghost trait, while the millions of other descendants in the colony completely lack it.
A ghost trait moving through the generations. That is wildly cool. Now, before we had cheap, automated genome sequencing machines, scientists actually exploited transduction to draw genetic maps, right? They turned this viral accident into a tool.
They really did. They relied heavily on the math behind generalized transduction. Think back to the P22 phage. Its rigid icosahedral capsid can hold a maximum of roughly 43,000 base pairs of DNA.
Okay, that sounds like a lot.
It is, but the entire Salmonella chromosome is massive, about 4.8 million base pairs.
Ah, so a single virus can only physically scoop up less than 1% of the bacterial genome at any one time.
Exactly. The probability dictates the map. If you have two bacterial genes that are located on opposite ends of the chromosome, the mathematical chance of both of them accidentally getting scooped up into the exact same 1% capsid is practically zero.
Right.
But if two genes sit right next to each other, they are highly likely to be packaged and transferred together.
The closer they are, the higher the, wait, what's the term?
Co-transduction frequency.
Co-transduction frequency, right?
Yeah. By running thousands of infections and painstakingly measuring how often two traits transferred together into a new cell, geneticists could infer physical distance. They used those frequencies to build incredibly fine-scale maps of bacterial chromosomes decades before sequencing technology existed.
Using a blind loading crane to do cartography. That is ingenious. Okay, we've explored the discovery, the mechanics, and the mapping, but we need to address the great misconception, because even mainstream popular science writing constantly trips over this next concept.
It is arguably the most pervasive tangle in microbiology literature outside of textbooks. This raises an important question about some of history's most famous bacterial toxins.
Yeah, the sources highlight diphtheria, whose mechanism was figured out in 1951, cholera, unraveled in 1996, and the Shiga toxin, found in highly pathogenic strains of E. coli that cause severe food poisoning outbreaks.
Right, very famous toxin.
If you read a popular science article about these diseases, the author will almost invariably claim that these dangerous toxin genes spread between bacteria via transduction.
And strictly speaking, using the word transduction in those cases is biologically incorrect. Those are actually examples of a distinct phenomenon called lysogenic conversion.
Lysogenic conversion. I'm struggling to see the boundary line here. Because in both transduction and lysogenic conversion, a virus lands on a bacterium and changes its behavior, right?
They both involve a phage. which is why reporters conflate them. But the origin of the genetic cargo is fundamentally different.
How so?
In transduction, as we've established, the phage is acting as an unwitting courier for bacterial DNA. The genetic material originated in a previous bacterial host. In lysogenic conversion, the toxin genes like the tox gene that causes diphtheria or the CAKX-AB genes that cause cholera, those belong natively to the virus itself. It's viral DNA.
Oh, I see the distinction. It's not an accident. The virus isn't carrying stray bacterial furniture it's scooped up in the dark. It actively brought its own heavy machinery to the site.
Precisely. The temperate phage infects the bacterium and integrates into the chromosome as a dormant prophagia, just like we discussed with specialized transduction.
Right.
But while it's sitting there in the chromosome, supposedly dormant, it actively expresses its own viral toxin genes. The phage's own code is doing the work to change the bacterium's phenotype. The bacterium isn't naturally toxic. It's just a puppet being forced to manufacture a viral poison.
Let me make sure I'm locking this in for you listening, because this is the kind of distinction that makes or breaks a biology exam. Transduction is when a virus accidentally moves bacterial DNA from one host to another.
Correct.
Lysogenic conversion is when a virus successfully expresses its own DNA to alter the host.
Exactly.
It's the difference between a mailman accidentally delivering your neighbor's mail to you, that's transduction, versus the mailman moving into your spare bedroom and blasting his own terrible music all night long, that's lysogenic conversion.
That's a very vivid way to put it. But yes, the mechanisms are entirely separate. One is a packaging or excision error of host code. The other is the deliberate expression of viral code. Microbiologists insist on keeping these terms cleanly separated. But because both scenarios yield the dramatic, clickable headline, "A virus gave the bacterium its poison," the nuance is routinely lost in translation.
Well, now you know the difference, which means you can officially spot the sloppy usage in the wild. So what does this all mean? Bringing it all back down to earth, why does this microscopic distinction between a blind crane and a panicking sleeper agent matter to human beings living our lives today?
It matters because of the terrifying math of the microscopic world. Bacteriophages are the most abundant biological entities on the planet.
Right.
In any given environment, soil, ocean water, hospital surfaces, viruses outnumber bacterial cells by an order of magnitude.
Billions upon billions of them.
Trillions. Every single lytic infection cycle, happening trillions of times a second across the globe, represents another opportunity for a packaging error. This isn't just an academic curiosity confined to a petri dish in 1952. This is a massive, ongoing engine for how bacteria swap survival traits in the wild.
Traits like antibiotic resistance.
Yes. If a phage accidentally packages an antibiotic resistance gene from a surviving bacterium, it can fly across the environment and deliver that resistance to a completely different susceptible bacterium. They don't need to physically mate.
They don't even need to be close.
Right. They don't even need to be in the same immediate microenvironment if the virus gets carried by a water current or on a speck of dust. Understanding transduction is critical to predicting how virulence factors and multi-drug resistance spread across bacterial populations without warning.
And it's not just happening randomly in the wild. We actively harness it for our own benefit, right? The sources highlight that phage P1 transduction is still a standard everyday bench technique in modern genetics labs.
Oh, absolutely. More than 7 decades after that filter experiment, scientists still rely on P1 phages. If you want to move a specific targeted mutation from one strain of E. coli to another, you grow the virus on the donor strain, harvest the newly built viral particles, and intentionally infect the recipient strain.
It's just so much easier.
It offers an incredibly clean, precise way to move a single genetic marker without the laborious multi-step process of chemical cloning.
We literally turned the blind accidental delivery truck into an on-demand courier service.
We did. And that leads into something truly profound when you realize the scale of this mechanism.
Oh, I like profound. Where are we going with this?
If we connect this to the bigger picture, think about the sheer mathematical scale of this in your own gut microbiome right now.
Okay, slightly unsettling, but go on.
With trillions of viruses and bacteria interacting, phages aren't just microscopic killers. They act as a blind, accidental internet, constantly shuffling the genetic deck of the bacteria inside you every single second. What if we take that on-demand courier service out of the petri dish?
You mean use it as a therapy?
Yes. If phages are constantly rewiring bacteria in the wild, it makes you wonder about the future of medicine. Could we one day engineer our own fleet of viral delivery trucks to rewrite the microbiome in our own guts?
Oh.
Imagine swallowing a pill full of custom phages designed not to kill, but to perform deliberate therapeutic transduction, fixing metabolic disorders or deleting antibiotic resistance from the inside out.
It completely flips the narrative. We've spent a century viewing viruses strictly as pathogens to be eradicated, but they are just vehicles. If we master the exact mechanisms of how they load and unload genetic cargo, the same microscopic accidents that spread disease could become the very tools we use to cure it.
Exactly. It's a total shift in perspective.
Something to mull over the next time you're recovering from a common cold and cursing the viral world. Thanks for joining us on this deep dive. Keep questioning the invisible mechanics around you.